So, hi, hello, and welcome again. I think everything seems to be working. Okay, again, a little bit of few organizational things that I have to do first. Yes, sound seems to be here. Hi, and welcome to another Saturday live stream. Microbehunter here. That's usually the way that I always say hello to you. And sound seems to be good. Let me quickly check. Video also seems to be working. Chat is working. My microscope is working. Yes, I think we're ready to go. Okay. First of all, thank you very much for joining in again on a Saturday to a microscopy live stream. Today's topic a little bit, maybe I would say a little bit unique, a little bit different. Maybe I decided to put some hair under the microscope. And yeah, I've come up with a couple of little experiments. I wouldn't say experiments maybe, but just a couple of demonstrations as well. And I would like to simply show you some hair under the microscope, not only my own, but I also got, thankfully, yeah, some of my students also donated some hair, some of their hair to me yesterday. So we're also going to have a look at that. Traditionally, it's always like this, that during the first 3-4 minutes, I just say hello all over the world. And by reading the chat, because it usually takes a few minutes until more and more people start to join in. So there's a hello from London, from Canada, from Georgia, from the United States, from Greece, from the Netherlands, from Ljubljana, and from Germany. Sound is good. And okay, here we go. Ready to go already. Yes, if you're new to this, please feel free to post questions or comments in the chat. We will again do a little separation of comments. So if there are some questions, microscopy questions that are off topic, I will try to answer them after 1 hour during the second part of the live stream. And if there are some on-topic questions, of course, I will try to attempt to answer them during the live stream. Okay, so let's get started. Why hair? Yeah, it's a topic that is not quite very common, I have to say. Normally we look at living things under the microscope, microorganisms, things that move, and I said, well, why not try something different today? And that what you see right now over here, this is actually hair in cross section. And how in the world are you able to get a cross section of the hair? I'm gonna tell you, you'll be surprised. So those round things here, right? These are actually like discs of hair. Hair is of course long, we know that, but there's kind of like a cross section, and there are quite a few of those. The question is, how in the world did I get those cross sections? No, it was not a microtome. I did not do that with a microtome. And yeah, this is basically from today in the morning. I was shaving myself. And these are actually, yeah, basically it's my beard. Yeah, and this is actually a very interesting way, actually so far the easiest way of getting proper hair cross sections. So we're actually able to see, yeah, how hair looks like on the inside. Okay, and what you can see here is that the hair in the inside has, yeah, it has a region that's a little bit different. It's called the medulla. Yeah, and you see the different layers, and that's called the cortex. Yeah, and yeah, there's also the so-called cuticle on the outside. It's the outermost layer, right? Of course, if you take a hair and put it directly under the microscope, you're never able to see it in a cross section like this. So I kind of like this yeah, perspective as well. Okay, so. The thing that essentially, yeah, I see that there are a couple of off-topic questions already. Thank you very much about, for example, if we're able to live without gut bacteria. I can talk about that a little bit later on. Okay, yeah, so all of those questions that are not directly related now to the live stream, please do feel free to post them, and I will try to answer them later. Yeah. So again, more and more people started to join in. Okay, from Spain, hello. Okay, from Australia, from Vorarlberg in Austria, Lebanon. Okay, so yes, that's basically. I want to show you something else right now. Many people have been asking me, what about the colors? I've got special optics on my microscope that kind of make colors like this. If you want to. Yeah, remove the filter, then this is basically bright field. Okay, so this is basically the natural colors, so to say, and yeah, and that's basically bright field. You can of course adjust the contrast here as well, let's have a look at this using phase contrast as well. That is again a different thing. Yeah, and in phase contrast we see that, yeah, It doesn't always look very nice, but in phase contrast, this is how it would look like, and it's also a very high magnification. I just want to show it to you because those of you who joined in a couple of weeks ago when I put some diatoms under the microscope, it really looked nice. But generally, I just want to show you that fairly large and thick specimens do not look very nice under phase contrast. Yeah? So I don't know if some of my students actually joined in. I told them that I have a live stream today. And the reason is because thankfully some of my students donated some hair. And let me adjust this again. And just would like to show it to you as well. Today in the morning, from my time at least, I prepared some microscope slides with those hair. And I consider them quite interesting as well. Yeah. And yes, I see again a couple of off-topic questions. Please feel free to to talk about those. So I'm going to show you a little bit what I got here. And this is how I collected it. I'm going to show you the desk view here. Okay. Yeah, by the way, these are all some of my hair, which I'll be looking at today. And look, I'm going to show you, these are basically 18 hair from my students that I collected yesterday. I asked them, are you willing to give me one of your hair so that I have a topic to talk about during the live stream? Yeah. So here it is. And I did prepare a microscope slide because, of course, looking at it like this is not quite as convenient. And what I've therefore done today in the morning, I kind of arranged them also in a line here. It's not a very nice permanent mount. You see there's the glue all over the place here, but it works. Okay. What I would like to do now is I would like to quickly show it to you also under the stereo microscope, because one of the things that I want to do today, and that's a little bit this learning, how do you call this learning aspect of this live stream, is not just that we look at hair, but I also want to show you how to actually do some measurements under the microscope so that we can determine the diameter of a hair. And I would like to also show you a little bit the diameter of a hair in relation to some onion cells. And I'm now placing this here under the stereo microscope. Okay, I'm zooming out here a little bit. I have to focus. I have to reach over. It's a little bit, yeah. And the reason why I would like to show it to you also under the stereo microscope, because it kind of gives you a little bit of a perspective. It's kind of on how thick the hair actually is. Yeah, generally, as you see, it's not the best. Here we go. That's actually one of those hair, right? However, under the compound microscope, everything looks significantly better. Yeah, you see, it's that we can zoom out yet further. That's the maximum that I can zoom out. It's a little bit blurry. Yeah. And of course, it is not entirely satisfactory, right? For this reason, I've prepared this microscope slide. Let's switch this off again to save a little bit of electricity here. And for this reason, I prepared this microscope slide. And let's just have a look at this first, because there are a couple of interesting things here as well. Okay, so what I'm going to do is I'm going to go again to the scope view. Again, these are for those of you who just joined in, what you see here, that's from the morning today when I had a shave. So I collected the little, how do you call this, the beard, right? So you see that essentially you could actually do a calculation of and measure out maybe how many fractions of a millimeter the beard grows every day. Yeah, so that's actually, you see over here, that is over here, that's the amount of since yesterday, right? Yeah, so that's something one could measure out. Over here as well, and you can also see over here in the beard that obviously some of them are darker than others because, of course, with age, hair starts to turn white. But let's put this slide now under the microscope here. So I'm going to go down with the magnification. You always want to do that first, and let's have a look here now at the hair of my students. I'm just going to put it now directly in bright field again. Okay, here we go. Yeah. I will also tell you a little bit of how I actually made it in an arrangement like this. It wasn't so easy, but we can already see at the lowest magnification. Let's turn off the arrow. At the lowest magnification, we can already see that the hair, obviously, not only has different colors, but also different thickness. Yeah. So we want to go up a little bit with the magnification as well. You see a few air bubbles. Yeah, I've used the PVA glue, and what I have done here is I arranged them, or I tried to get them as parallel as possible. I cut off a little piece of hair and I dipped it in the glue and I kind of glued it first on one side. Yeah, and only after the glue has dried, this is when I applied the rest of the mounting medium, the rest of the glue, and then put a cover glass on top. Yeah, but when you go up, let's go up a little bit with the magnification. I always have to refocus a little bit, so maybe the whole thing is not properly adjusted. Here we go. Okay, so that's the 10 times magnification. Turn it like this. Yeah, and let's go up yet further. And let's go up yet further with the magnification, so 40 times. I'm going to open this up here a little bit, and what we are already able to see a little bit, and this is depends a little bit on the darkness or brightness of the hair. This is of course the surface texture. Yeah, hair obviously is not entirely smooth, yeah, but has a certain surface texture on the so-called cuticle, and that is also something that we're able to see. Yeah. The question that, yeah, so let's have I'm just going to read off a little bit some of the questions here. Okay. Yeah, so hello again from Arizona. Yeah. And yes, show a red hair. I don't think I have a red hair, unfortunately, but maybe I can organize one. I have to ask some of my students. I don't know if any have red hair. With dark field, do you think we can see real hair color? Well, let's put it this way. What you see right now, of course, with the blue background, that is because of the filters that I use. When I go into bright field, and I can try dark field as well. Okay, so this is bright field. That's the way that you would normally see it, and I would probably say that this would be kind of the real hair color. Okay, of course, you see as you change the brightness around, yeah. Of course, all the colors also change. But the only thing is I do have dark field, but only with the low power 4 times objective. Okay, so I can do that. So let's flip this one in here. Okay, I have to open it, and this here would be now dark field. I have to also refocus again here. Okay. And in dark, unfortunately, I don't have dark field with yet higher magnification. Let me try to, maybe I can get the, closing the field diaphragm is also not the way to go. Okay, here we go. And what you're able to see here is the following, something very typical is that at the edges look much brighter, right? And this is because the light that strikes the edges, so also here you see it quite well, is basically, redirected into the objective. Look, this is actually quite interesting. I'm just noticing this right now. Do you actually see that here the right side is brighter than the left side of the hair? What I'm going to try now is I'm going to try to move now the hair over to the other side. Look at this. Isn't this cool? Now you have to check a little bit. Yeah, you see? And the hair now, follow this here. Now the hair is brighter on the left side compared to the right side. Yeah. So you can actually see that if you use a dark field, then you can also, and you're kind of moving the specimen left and right, and the light either strikes it a little bit more from the left or more from the right. This might remind some of you of so-called oblique illumination, where this is actually done, over a much larger area as well. Yeah. So there is, interesting, some comment here. There's a fun fact. I'm just reading off the comments right now. There exists something called the beard second, which is the distance a beard grows in a second and should be about 10 nanometers. The definition was inspired on light years. That is interesting. Yeah. I am, next question here. I'm African. Would my hair look different in comparison? And look, I'm going to tell you the following. You should be able to, see here that I'm not forgetting about the question. I'm not forgetting about the question. I'm not forgetting about the question. Look at this. How different the hair look over here. Yeah. And I've got students from all over the place, right? So would the hair look different? Sure, because different people have different hair. Yeah, for example, in my case, I'm going to show you my hair later on as well. I've got a black hair, right? Um, uh, um, and also fairly thick hair, so you're going to see that actually my hair again looks different to the hair of the students that I have here. Yeah. So of course, it's we see. However, I would also say that let's not forget that even from our own body, hair from different parts of the body look different. So for example, the eyebrows and the hair here and the hair, the fine hair on my skin also look different. Yeah, so there is a huge, how do you say this, a spectrum of thickness, for example. Yeah, but you know what? I cannot help it. I cannot help it, guys. Just give me a second. Yes, this is one of the nice things about this live stream. Things like to be, I like to improvise. I want to know it now. Okay. So let's do the following, because there was this comment of a beard second, and somebody please do the math for me. Then look, let's do this a little bit like this again, so that we see it a little bit better. I want to measure out now. Oh, beautiful! Ha! Very nice, very nice. Look, I'm going to show you something. We are very lucky. We are very lucky. Do you see this here? Actually, over here, this beard is right on its edge, right? It's right on the side. This is seen from looking into the beard, right? That's the so-called the medulla, the central part. And here we're lucky that this is right, seen from the side. So this is basically the amount that my beard has grown within 24 hours, because I shave myself every day, usually at the same time in the morning. Yeah, and I know that. Look over here, it's a little bit larger. I get it. Yeah, so maybe, I don't know, maybe I pressed a little bit harder or less hard. Yeah, but who cares? Just as a how do you say, as a reference, from here to over here, this is in 24 hours. The question is now how much is this? Okay, so what I have here, luckily I'm going to show you the desk quickly. Well, look, some of you might already know this is a micrometer slide, and this basically has a scale on it. This is actually something I did not plan to do today, to actually measure out how much my beard is growing in a day, but why not? Okay, we can be flexible. And what does it say here? One division is 0.0 whatever. I cannot read it. We're going to have this, we're going to have a look at this, okay? And the thing is you know what I need to do? I need to somehow, I just want to estimate this. Is there a possibility for me to copy this arrow maybe? Do I have a second arrow? Yes, I've got two arrows now. Do you see this? Okay. So from here to over here, it's diagonal, so I have to actually also put the arrows diagonal. So from the tip of the arrow to the tip of the arrow, this is within 24 hours. You know what I think? I think I should go up with a magnification. This is not precise enough. So look, let's try this again here. Okay. So here we go. I think that's much better. So from this point to this point, this is now in 24 hours. The growth of 24 hours. Now let's see how much this is. I'll put the micrometer slide now in here, and let's see how much this is. Okay, I have to find. This is now the challenge. I have to kind of find this. I have to find this thing. So I need to go. This is the 20 times objective. So I need to find the scale. We got to be a little bit patient now. Up here is something. So it's upside down. One division, one line is 0.01 millimeter. So 0.01 millimeter is 10 micrometers. So I have to find this now. Where is it? Where is it? Where is it? Where is it? Okay, here we go. Here we go. Yeah, so here we see already the scale. I was doing this at 20 times magnification. Oh, I lost it again. Always center it before you, and focus before you go up with the magnification. That's rule number 1 in microscopy use. So here we go. Over here, this is basically, look, this is basically now, I'm at 20 times. So from one line to the next line is 10 micrometers, which is approximately the size of a human cell. Okay, this means we have how many divisions? One, I have to count them. We've got basically over here to over here, 1, 2, 3, 4, about 5. 5, yeah? So 10 micrometers by 5 is 50 microns, 50 micrometers in 24 hours. Did I get this correct now? Yes, 10 micrometers is 1 division, times 5 is 50 micrometers. Okay, so that's basically, the growth of this one here, was 50 micrometers, in 24 hours. Okay, and then we can now divide the 50 micrometers. Somebody do that. Some 50 micrometers is 5,000 nanometers. And then you can divide it by the seconds in a day, and then you know how many nanometers per day the beard is growing. Okay, I'm getting sidetracked right now. Yeah? So I'm going to read again some of this. I'm going to go read some of the comments again. Okay. Great to catch the stream before the Northern Lights. Oh, okay, very nice. You've got Northern Lights. So what am I doing today? Somebody joined in later. Today I'm putting some hair under the microscope and just measured out how much my hair or my beard was growing in one day. Yeah, it's very quickly, huh? Could you identify somebody just by the hair? Well, I think yes and no. You see, nowadays you probably... Well, I heard that yes, it is possible to kind of, just looking at the hair texture, if you have some kind of limited number of people to choose from, right? Then it might be possible maybe to allocate whose hair it could be. I don't know, let's say you've got 10 people, they have different hair, right? And then maybe you have a hair sample, maybe you can compare it and you can reduce it. But generally nowadays, if you have a hair sample, you're probably going to do a DNA analysis in forensics. Right? But you always need something to compare it to. Yeah, that's the thing. Yeah, if you don't have a reference, then you won't be able to know who it is. Yeah. So I have a micrometer slide and never used it. Okay, yes. They're actually quite... I'm going to show you later, if you do not have a micrometer slide, I'm going to show you later another method on how you can actually make measurements. Okay? Yeah, so that's actually another point that I would like to do today. Yeah. My arrows are diagonal, so it's more 7 to 8. Yes, that could be. Yeah, that's correct. Yeah, but considering the fact that the whole thing is kind of, how do you say, not very precise anyway, because depending on how hard I push when I shave myself, of course the lengths are a little bit. That's one of the things that are a little bit would be also kind of nice if you somehow were able to kind of rotate the stage as well. Yeah. So in any case, that is basically very briefly a measurement of my. And let's go quickly back to the to something else that I would like to show you. So let's take this out. Let's simply remove the beard again, and I would like to show you now some of my hair, which I basically also made a permanent slide. I prepared this because it's simply faster this way. Okay. And this is actually quite I think I can remove the arrows again here. So let's just take them away again. Okay. And here it is again. Now that's a dust fiber. This round thing that you see over there, it's dust. This here is a hair. And here we already go. That's exactly the place I wanted to show you. This is a pretty coincidence. This is basically both of my hair. Yeah, and what do you notice? You notice that I'm already becoming old because this is actually one of my hair that actually turned gray. Okay, so where is it? Yeah, so that's one of my black hair. And this here is one of my gray hair. And what do you notice? You see that the inside here is pretty dark. That's the so-called the medulla on the inside. And here it is pretty dark, but if you follow it around a little bit, I think it's not dark everywhere. See, it's starting to become light over here. Yeah, you see this? And that's of course interesting. So what in the world's going on here? And so there seems to be, I don't know if it's just the pigment or so, but it also What I also heard is that sometimes the medulla, the inside, could be actually hollow. So my first idea was that maybe there's some air inside here, and this causes a huge difference in the refractive index and maybe causes it to appear black. Yeah, the inside. Yeah, but it doesn't really look like air either. So there still seem to be probably some pigmented cells in here in the center. Yeah? But my first impulse was to think, wow, there must be some kind of air in there, which makes it kind of dark. But this does not look like air bubbles, really. Yeah. So sometimes when you have kind of empty spaces somewhere, and when it fills up with air, then it looks quite different from the surrounding. But here, this doesn't look like air, right? But still quite nice to see that that the hair has different layers. Yeah, that's basically what we have here. Yeah. Is it possible to take measurements using the cross table? So the question, I think, I don't know what you mean with cross table, but maybe you're referring to the stage micrometer, the scale that you have on the microscope stage. Maybe that is the thing that you're referring to. I'm going to show you just a second. Look at the top. Okay, look at the top. What I'm doing. Maybe you're referring to this scale over here. Right? And yes, it is possible not to make very precise measurements, but it is possible to make measurements with this as well. Okay, so basically what you're doing is you're kind of remembering a point somewhere on the slide, and then you're kind of moving it, right? And then you're reading it off again, and then you actually know how much, you've moved it, and then if you've remembered a certain position, then you have actually also determined the size. Yeah. So this stage micrometer can also be used to make very rough estimates, but it's by way not as precise as using a micrometer. Okay, if this is what you mean with cross table. Yeah? Can you show a hair follicle or bulb? Yes, I have got a permanent slide, a very nice one that I would like to show you as well. Yeah? Does the medulla determine the hair color? Not necessarily alone. I'm going to show you why. Because apparently, even though there are a lot of pigmented cells in there, as we can obviously see, just want to show you. I need to make this darker again a little bit so that the whole contrast is better. I have to find some of the other hair. Look, it's interesting. Over here, it's completely white. Yeah, let's go down. It becomes darker, darker, darker. But look, that actually the whole hair is, this is one of my regular black hair, right? It's actually not the inside, but it's actually the whole hair that has the hair color. Yeah, and look at this, how different it's basically from my hair. Yeah, but look how different the thickness is. Yeah? So this is something I just wanted to kind of show you, because I found this kind of remarkable a little bit, this big difference between gray hair, the few gray hair that I already have, and the black hair. So the contrast is quite intense here, right? So this is and what I want to quickly determine is the following. What time is it? I'm trying to always make a little time estimate. And I would like to do, and I've been thinking a long time, should I show it to you or should I not show it to you? There is a little rule that as soon as you start doing a little bit of math, people start clicking away, but don't worry, maybe just a little bit, very little, okay? I want to show you how to do size calculations using a ruler and a microscope. A ruler like this. Yeah, it's possible. You're actually possible, and I just thought I'm just going to show it to you. Okay, but it's really not a lot. But I know that it's not everybody's interest. But I think it's very nice because how do you say? Everyone's got a ruler at home. Yeah, and you can actually do pretty good measurements. And I would like to show this to you. And in order to do that, we're going to do the following now. And I'm just going to put and I prepared something already because I cannot show it to you on the monitor, but therefore I prepared it for on the, how do you call this, on paper. Let me quickly close this here, and let's put this out here, and let's take this out here. And I'm going to put the ruler now directly under my low, look at the top. This is the low, how do you call this, power objective. Okay, so, and you might want to focus a little bit. And then what I've done is I've taken a picture of this, right? At the lowest magnification, at the 4 times objective. Why at the lowest one? Because you can then see the millimeters, right? If you go up too high with the magnification, you cannot see it from line to line. Maybe I can still do that. Yeah, barely. You see, I'm just able to get 3 lines, right? Because I also have a very wide field of view. So generally, what I always say is to take a picture with the 4 times, right? And this is what I have done. Look, I'm just going to show you. Look, okay. I've simply taken a picture of this, and then what I have done is the following. I've taken a picture, and I'm going to show you now the whole thing using the desk view. And yeah, so that's basically what I've got here. Let me move the camera up a little bit here so that you see it better, right? So I've taken this the picture of the ruler with my 4 times objective, right? These are scratches on the plastic, all right? And then what I've done is because the hair is so much thinner, what I have done is I've taken a picture with the 40 times objective, right? And you see that's exactly the place that we just had a look at, yeah, yeah. And then I printed it out in the same scale, right? So I did not change the magnification, and later on I just used the different objectives and simply printed it out, right? And now what I have now is I have a reference here, a millimeter, and I can do a little bit of math, and then I can calculate how thick my hair is. Yeah. However, the problem is look, I've taken it with a different objective. So I would like to show you now a little bit the math that I've done. Okay. I'm going to. It's actually quite easy, and it works like this. You take a ruler. So I need the ruler again. And what you do is the following: is you measure out the diameter. Okay, I need to move this over so that you see this better. Okay. I have to measure out the diameter of the hair. Okay. As it is magnified, and that is 53 millimeters, and I've got here, yeah. So that is, I'm used to the metric system, yeah. So it's 53 millimeters, right? And that's basically the thickness of the hair magnified with a 40 times objective printed out, right? And the thing is now the following. Over here, I've got the 1 millimeter. But you see that I've used a different objective here. So this one over here is magnified 40 times, is 10 times as much as 4 times. So I cannot really compare it, right? So what I have to do is I have to divide this by 10, the 53 millimeters, so that the 2 pictures can be compared. So what I do is the following. The 53 millimeters, I divide this by 10. Yeah. So I have then 5.3 millimeters. That would be now... You cannot see it, okay? This would be kind of the value that I'm using for doing the math, right? So, and then over here, I do the following. Now the 5.3 can now be compared to the 1 millimeter over here. And now you see that the lines here are pretty thick. So actually, what I recommend that you do in this case is that, let's say that you take the left corner here, and you measure the left corner to the left corner. Yeah? And in this case, it's exactly 50 millimeters. It goes all the way exactly to 50. Yeah? So that is yeah, another value that we have to remember. It's from here to here. It's from here to here. I can write it from here to here. It's 50 millimeters. Okay. So and now what I can do is I can do a direct proportion calculation, which I'm going to show you. I don't know. I need some kind of a piece of paper where I can do a little bit of math. Right? So, I'm going to now do the following. Okay. 50 millimeters, okay, on the paper here corresponds to 1 millimeter. Okay, because that's in real life, this is 1 millimeter. Okay. And now I move over here as 5.3 millimeters. 5.3 millimeters corresponds to the actual diameter of the hair, which is X. It's direct proportion calculation, right? So over here I have the enlarged image of in the picture on paper, and over here I have the real size. And again, what I have done is I had to divide it by 10 because I used, because the 40 times objective magnifies 10 times more. And then when I have this here, what I can do now is I can cross multiply this times this divided by this. Yeah, so 1 times 1 millimeter times 5.3 millimeters is 5.3 millimeters squared divided by 50 millimeters. Yeah, so it would be 5.3. Yeah, this times this divided by 50. Okay, and I have done the math already before. Okay, yeah, so basically that's 0.11 millimeters. 0.11 millimeters, which is a tenth of a millimeter. And this basically, let's do a common sense check. We just take 10 here and put it next to each other, and then it gives me actually the diameter of 1 millimeter. Yeah? So the common sense check also makes sense. So I can basically, I've determined that this here over here, yeah, this here over here is approximately a tenth of a millimeter in diameter. And I was able to calculate that without using a micrometer slide, but simply by using a regular ruler, which everyone has, and a little bit of math. Okay, and if this was a little too fast for you, then don't worry. The video will be available online afterwards, and you can always watch it again. But I just thought that I want to show it to you because I'm actually using the same technique also with my students in school. Because you can essentially, measure out pretty much everything using just standard, readily available equipment. Yeah? The question is how thick is the medulla? Okay, that's an interesting one. It's actually quite, that can be easily determined. So if this one is 53 across and the medulla is over here, let's say 12 millimeters across, okay? So that basically means 53, well, let's stay with this here. If 5.3 millimeters, let's make it easier. Okay, let's say 53 millimeters on the sheet. This corresponds to 0.1 millimeter. Okay, this here is 0.1 millimeter. Then 12 millimeters. 12 millimeters corresponds to x. So we may multiply 0.1 times 12. This basically makes 1.2 divided by 53. Gee, I don't have a calculator, right? We basically again cross multiply this times this divided by this. Yeah? So it's 1.2 divided by 53, and this is basically then the diameter of the medulla here. Yeah? Actually, we can actually, I don't know. Yeah, I don't know. It's 1, 2, maybe. It's approximately a quarter to a fifth, of the medulla here. It's an interesting thing that I found out, that you can actually determine in many cases whether the hair that you're looking at is human or not, because generally the size of the medulla in human beings is smaller than in other animals. Yeah, so if it's around a quarter to a third maximum, then it's probably human hair, and many animals apparently have a medullus that are significantly larger. I just read somewhere. So that's actually also quite interesting. So let me have a look here. So there is I hope I'm not overlooking anything. On your picture of the scale of the ruler, we can see an interesting regular pattern of the ink. Yeah, yes, exactly. Yeah. Okay. Another thing that I just want to say is the following. You see, concerning the precision, you see the hair is also different. Some is thicker, some is thinner, right? So even if we now basically have a very precise measurement on the thickness of the hair, maybe the next hair that I'm going to look at is again going to be different, right? So if you want to be scientifically correct, then you have to actually measure the diameter of many hair, and then you have to calculate an average and also standard deviation. Yeah, and so I'm just saying that there is no point in adding a lot of decimal places because the next hair that you're going to look at is going to be again different anyway. Yeah, so that is just a comment that I want to make here. Yeah, so yeah, I just wanted to show this to you because yeah, it was a little bit I don't know, I at least consider it kind of yeah, interesting, relevant, I don't know, yeah, to kind of use a regular ruler also to make measurements under the microscope. Yeah, so I want to move on. Okay, checking the time, 20 minutes. Look what I've done here. Next thing that I want to show you. Look, you've seen this already. That's my hair, right? You see that a little bit. It's yeah, white. And look what this is. This is also my hair. Why is it so much brighter? Let's move it down here. And you know what? Not only here. Why not just put it under the stereo microscope? I'm gonna tell you I've done something to the hair to make it brighter. Here we go. No, that's the one here. So yeah, let's turn this around here. Here we go. So let's go up. That's already, this is already the lowest magnification that we can have. So that's the original hair that I cut off. You see that there are some individual hair. Yeah, are still kind of white. And look at this guy over here. What in the world happened here? Why is it brown? It's my hair. However, what I've done today in the morning, I added hydrogen peroxide, H2O2. Some of you remember it. We used it a couple of weeks ago when I prepared the diatom slides, right? And the hydrogen peroxide is highly oxidizing, and it reacts with organic materials, and it started to bleach the hair. Yeah? So this is kind of bleached, right? And just wanted to show this to you, the effect of hydrogen peroxide on hair color. Apparently, yeah, that's why some people apparently use hydrogen somehow, or hydrogen peroxide containing things to bleach and to make the hair brighter. Yeah, yeah. Hydrogen peroxide is H2O2. I used 30% hydrogen peroxide, and you do not want to mess around with it. I have to give you a warning. This stuff is very aggressive at 30%. 3% hydrogen peroxide can be found in disinfectants, hand disinfectants, not a big issue. You still don't want to get it into your eyes. But I got some 30% hydrogen peroxide today on my finger, and it kind of burned my finger. At least didn't burn it. The top layer of the skin actually turned completely white because it's highly reactive to hydrogen peroxide. Yeah? So I just want to warn you a little bit, but I simply wanted to demonstrate to you a little bit what happens when people bleach their hair with oxidizing agents because it turns the hair brighter, right? And now you can see that over here I've got this brown. I don't know. Should we put this under the microscope as well? Why not? I'm just gonna make. I've not tried this. I'm just gonna make a very quick slide. Why not? Let's just compare them, just for the fun of it. I need some tweezers. Where are my tweezers? Where are my tools? Here they are. Where are my tools? Yeah, it's a little bit easier to take out the hair. Yeah. If you have problems, and that's a general comment, if you have problems adding water to your sample because your sample is too hydrophobic, and animal fur, for example, is a case here, then you should use oil. Yeah, but this one is fine because this one is, yeah. So I know this is a little bit sloppy to have them both next to each other. I know. Yeah. I usually do the following. If I were to add now a little bit water right now, then there's the danger that the whole thing is going to start swimming away. So what I usually sometimes do is I add a cover glass. Look, this is a pretty large cover glass. Yeah, I usually add the cover glass directly here on top. Then I take here is a little bit of water, and then sometimes what I do is I simply add the water here from the side. Does this even go in here? No, it's too high. Okay. Demonstration failed. Okay. This is here. Now we go. So yes, now. Because sometimes I tried this and then kind of the hair started to kind of float away. I think that's enough. This is a very quick and simple preparation. Let's have a look at this under the microscope, how hydrogen bleached hair and regular hair looks like. Here we go again. Let's see. Put this away here. It's too bright. So this one is now the hydrogen bleached hair. I should probably adjust the microscope a little bit better that I do not have. I'm losing part focality all the time. Yeah, and I think it looks kind of nice. Yeah, because you can see more details. Yeah, and that's looks kind of pretty. Huh? And let's quickly move over to the black here. Yeah, and it's so dark and so pigmented that actually we're not able to see any details or almost no details at all. This one here is a little bit brighter, yeah, but most of them here are quite dark. Here it's also a little bit brighter. Yeah. Okay, some air bubbles, and in comparison again, this is the one that I left in hydrogen peroxide for a couple of hours. Yeah. So I'm going to read again here. Okay. My professor once said reactive oxygen species like H2O to react with everything they should not react with. To this day, I love this quote. Yeah, that's the thing. H2O2 is very aggressive. It's unstable, obviously, and it really likes to react with organic substances. Yeah, I think I remember a couple of years ago I did an experiment like this, and I left the hair in there for much longer, and then actually not only turned white, but actually it started to dissolve. Yeah, obviously, because the organic material, the protein, kind of also got oxidized, and the hair started to become thinner and thinner, and until it kind of started to become completely dissolved. Yeah, how would you adjust parfocality? The thing is the following. My microscope is parfocal, but it's not properly adjusted with the camera. I think that's the problem that I have right now. So this is the thing, and I have to simply adjust the distance of the camera, and then it's gonna work again. Haven't done this yet. So that's basically something I just wanted to also show you a little bit. And another thing, because this has been a request, I want to show you now some permanently mounted slides, because I would like to show you now some so-called hair follicles. Again, and the following slides I did not make, but they're pretty good. Maybe because I didn't make them. And this one over here is, I can show you. This is actually quite a nice one here as well. It's a histological slide. It is basically the scalp of a human being. So this is actually human tissue here. It's a commercially prepared slide that we're also using in education. I remember that somebody once emailed me. He or she bought a set of microscope slides, and it was labeled with human. There was human blood and human tissue on it, and this person was a little bit worried. I said, "Oh my gosh, this is actually human tissue on here." I said, "Yeah, of course." This is quite common in the medical field, right? But the thought alone was not very comforting for the person. So I'm going to switch over to bright field. Okay, and the colors that you see right now over here, actually because it has been stained. So it's been commercially stained. So that is actually the way that it looks like. And I'm going to try to find a good sample here. These are all hair follicles, the red ones, but before I start explaining this, I would want to find one of them that's a little bit better, because they look very incomplete. I will explain why they look so incomplete. Yeah. Because they have been cut. Yeah, here we go a little bit. Look, this is a hair follicle in cross section. I want to explain this as well. Let's go up with the magnification here. Let's go up. Okay. This is the hair follicles can be found in the skin, and they produce the hair protein, because hair is mostly protein. Yeah. And look at this here. It's the arrow over here. This is a cross section of the hair follicle, and this dark red thing here, that's the hair. right? And do you see those little things here? Those little, these are the cells around it that actually make the hair, yeah, in the hair follicle. Yeah? And this dark thing here, that is the hair in cross section. Yeah, so that's yeah. And what I would like to show you somewhere here as well. And you can see that yeah, lots of there was one of them that was a little bit better, and I have a problem finding it now. Yeah, this one here, these larger ones. Yeah, that's the one I wanted to show here. Here, that might be the one. Yeah. Look, it looks very long, and the reason why is because when they cut the tissue, it was cut diagonally, right? And what you can see again here is you can see here a lot of cells, yeah, and over here where it looks a little bit like fibers, I think this is actually where the hair is, yeah, is made. And it's that long because the hair follicle was not cut all the way, but it was cut diagonally. So if you essentially, I don't know, let me use a pencil here as an example. If you cut the pencil diagonally across, yeah, then essentially you're also going to see that it looks oval, yeah, and elongated. Yeah? And that's basically what we see here as well. Yeah? This one here looks very much like a blood vessel over here in the tissue. Many of the cells in here that are so white very often are adipose cells, basically fat cells. They do not pick up the stain very well. Yeah. So essentially this is maybe it's better to go down with the magnification a little bit because it simply gives you a better overview. Yeah. And yeah, here basically is another one of these yeah, here follicles, yeah, and with hair being formed on the inside. And I think that these things here that you see standing out here are actually part of the hair protein. Yeah? And the red ones are the cells that surround the hair that actually make the hair. Yeah? So I like this slide because it, yeah, it's not only nicely stained, but it kind of also illustrates this. Also very nice. This here it looks, I don't know, like these round, yeah, these seem to be oil glands. Yeah, they have this winding, configuration, right? And those oil glands often they dump the oil and the fat, of course, into the hair follicle. And yeah, also over here, I think you see right here at the base, these seem to be some glands, sebaceous glands, yeah, that make oil. So, and this is a nice one. This is a nice one. Okay, folks, this is really good. Do you see this here? This long thing here connecting to the hair, right? Or to the hair follicle. These are actually muscles. And what you have is many hair are actually connected to muscles because when you're feeling cold, then the hair and you're getting these goosebumps, yeah, and then basically the muscles, they're pulling on it and this causes the hair to rise up. Yeah? So that means that yeah, many here are actually connected to muscles, and we're quite lucky that we're able to see this here as well. Okay, yeah? Those long muscle fibers that connect to the hair. Yeah? So let me quickly have a look here again at some of the questions. Off topic, permanent slides. Michael, can you explain why cut bubbles when you put peroxide on it? Can you please clarify? I do not fully understand the question. Donald is asking the question. Can you explain why a cut bubbles when you put peroxide on it? Do you mean basically why bubbles form when I think you mean to say because when you put peroxide, hydrogen peroxide on a sample, then it forms bubbles. These are oxygen bubbles. I will explain this, okay, later. It's a little bit off topic, and therefore I would like to collect all of those questions for later. Otherwise, I don't have anything to talk about later on. Okay, so that's one slide that I wanted to show you. Oh, okay, it's 53 minutes. I'm going to show you now another slide as well. Also a slide where I already made a video before, which you can find, of course, also in this channel. And this is, actually I like this slide even more. And this also, we're talking about here today. So what I have here this is an antique slide. And I don't know if you're able to read this, Tale of a Monkey. Yeah? I've basically made, this. I bought this slide some time ago, and this round thing that you see here in the center is the cross section of the tail of a monkey. I made a video about this. You can actually find it in this channel. It's one of my least liked videos, unfortunately. I don't know why. I thought it was kind of interesting, but not everyone probably appreciated the thought of looking at a monkey tail under the microscope. But the reason why I want to show this to you is because we're also able to see the fur, not very often, but also some of the hair of the monkey, some left over. And what we have to do is I give you a quick overview first, right? I give you a quick overview first. I'm not going to go into too much detail, okay? Because I made a separate video of this already. But yeah, this is basically how it looks like. Yeah. So in the center here, we've got the bone of the tail. We've got muscles here, 1, 2, 3, 4 muscle bundles that are pulling the tail left, right, up, down, right? But what I would like to focus your attention to are those things that you see over here. This here, all these long things here. These are the follicles that actually produce the fur. And you can actually see that actually some of them actually go outwards. Yeah. So this is actually the thing that I also want to show you. And this is again a little bit so this is again bright field. Yeah, and here again, and this is actually a very nice one over here where the arrow points to. You can actually see the hair, the fur growing out. Yeah. So, and when we look at it, we're able to see that it actually looks obviously very similar to our own hair. Right? Let's try the following. I think if you put on polarized light, then it actually looks nicer. Yeah? There seems to be, you see, it's interesting that hair seems to interfere and interact with polarized light. And and I think this has to do something maybe with the regular pattern, how the protein fibers are arranged, that this kind of has an effect on polarization, right? But that's simply also something I wanted to show you here. And over here are again some more of these here. And I just hear that I'm now receiving again a couple of questions. Thank you very much. Okay, I'll in just a minute or so I'll answer your questions. Yeah. So yeah, but I kind of just wanted to share this as well. Yeah. Yeah, there's a question about xylene. Yeah. Okay. Yeah. The monkey tail was like an all-in-one specimen. Yes, that's correct. Because this monkey tail specimen really contains not only bones, not only connective tissue, it contains also follicles, it contains muscles, it contains blood vessels, right? All sorts of things. If you're interested in this, yeah. You can watch my video. But I just wanted to show this to you here as well. Here, for example, this one over here, what you see is an empty hair follicle from the fur. So, what time is it? 57, yeah. I will do the following. What else? I don't know. I actually wanted to do the following. You know what? I'm going to do this now still because I planned to do it. Because we have been talking about the size in micrometers and so on, the math part. But you see, what I'm going to do, let's compare the size of hair, the diameter, with the size of a cell. Okay, because this kind of illustrates a little bit how big cells actually are. This is actually something I wanted to do and plan to do, and so we're not going to skip that. Yeah, I'm going to simply make this the standard onion slide, like many of you probably already know how to do, and if not, then it's also good that you're able to see this, what I'm doing right now. And let's hope that we're able to find this thing here. Look, this. Did I get it? Yeah, here it is. Okay, do you see this onion skin? Let's put it over here on the slide. No water yet. Yeah, I'm going to put this away, and the whole room starts to smell like onions. And I'm going to put a little bit here, one of my hair, a few of them, because that's one of the problems with microscopy or biology. Of course, we have all of these nanometers and micrometers and I don't know what. but sometimes it helps to make a direct comparison. Let's see how the size of the diameter of a hair is in relation to the size of an onion cell. That's kind of the point. Yeah. And we put a cover glass on top. You know what? I put it directly on top, and then I want to add some water, of course. Okay. Let's add a little bit of water. Yeah, it's just enough so that the capillary action allows everything to stick together. And let's have another look here. Where is it? Here it is. Here we go. And you're going to be pretty surprised. You're going to be pretty surprised because I'm going to explain what you see now. You see this black line, obviously that's my hair, okay? That is my hair over here, right, obviously. And you see those things here? That's a cell, that's a cell, that's a cell, that's a cell over here. This basically means those onion cells here have the same thickness approximately as the width of my hair. Those onion cells are pretty large. Obviously, that's why they're such a popular specimen. But I just wanted to show it to you that, yeah, if you want to know how big is a cell, yeah, they're kind of small, they're tiny. Yeah. And I say, well, yeah, in many cases they are indeed are. But some cells like those onion cells over here, look, they are pretty much the same. They have the same diameter as the diameter of one of my hair. Okay, just wanted to show this to you a little bit, yeah, that sometimes some cells are probably not that small as we might think. Obviously, the cells that are much smaller, right? But those onion cells are pretty huge. And as a matter of fact, you can even, if your eyes are good and if you take a good magnifying glass, you're actually able to see those cells also with a magnifying glass. Yeah. Just wanted to kind of illustrate this a little bit so that yeah, you see a little bit the size relationship. Yeah, and you can also barely see that the inside here is a little bit darker again, so you see again the medulla. Yeah, yeah. So I think that's kind of nice. Yeah, over here as well, you see some more here. It's coincidence that they're basically now running into the same direction as the cells, right? Yeah, yeah. So. And I'm now using my 20 times objective. Yeah, so this can be done quite. The round things are of course air bubbles. Yeah, and maybe just because we've got onion cells now here under the microscope, let's do the following. Now let's make it brighter here. I don't know if you're able to see this. It's difficult. Yeah, you got to be really super patient. But sometimes you see those tiny dots. Maybe I have to open this a little bit. You see these are vesicles, and sometimes if you those tiny dots, I don't even know if you're able to see it properly over the live stream. But these vesicles actually move around and yeah, transport substances inside the cell. Yeah? So I made time-lapse videos here. Yeah? So and yeah, I just wanted to show this to you. And now what I'm going to do is I'm going to. I don't know, I guess I'm just going to switch over and I'm going to continue, and I would like to answer a couple of questions that you had. And yeah, I see that some of the questions, yeah, if there's some more questions coming later on in the live, in the chat, then I'll talk about those as well. But these are, and now I'm looking at them for the first time. This is usually always the scary part, because now I have to come up with some good answers, and sometimes I obviously didn't prepare. Yeah, some off-topic questions. Can we live without our gut bacteria? Gut bacteria are bacteria in our digestive system. And indeed, some weeks ago, I did some research on this because scientists have made mice and rats which are bacteria-free. And basically, apparently they were also able to survive. They were not able to digest food as efficiently. And the thing is the following: we're not able to test that on humans, because we human beings obviously have bacteria in our digestive system, and they play actually very important functions, because those bacteria provide enzymes for helping digest food. However, in some animal experiments, they have found out that even bacteria-free animals, which apparently they were somehow able to create, were able to survive. So this is the question: to what extent those animal experiments can be transferred to human beings. Generally, I would say that I mean, we humans are adapted to our environment, yeah. And if you make big changes, yeah, we have more bacterial cells growing on our body and in our body than we have body cells. So they're symbionts, and I would probably say that they're pretty important for survival. But then again, there are some experiments that they've made on laboratory animals which were able to survive without those bacteria. Yeah. Off topic, I'm thinking of starting a reef tank with live rock. So we'll have microspecimens from the sea. Many years ago, over 20 years ago, I did myself. I had a freshwater aquarium, not a saltwater one, but a freshwater one. Yeah, it was a very nice hobby. And right now, I don't have an aquarium anymore. It's also a space problem a little bit in my living room. But I do keep some water samples inside a jar. Yeah. So this is actually quite, yeah. Quite a good thing. If you it can be combined, microscopy and keeping aquariums, home aquariums can be combined quite nicely. Yeah. With dark field, do you think we can see a real hair color? I think I just talked about that. The thing is the following: that any kind of it's a somewhat a philosophical question. Any kind of technique in the microscope, dark field, DIC, polarization, even bright field. Because it was bright field, you can also adjust the contrast and so on. Any type of technique with a microscope changes the colors somehow. So the question is what is now real, right? If you want me to go philosophical, what is my real hair color? Well, it's black. Well, actually, no. It depends a little bit how the light shines on it and the brightness of the environment and so on. So even, yeah, it's a philosophical question. What is real, right? Basically, generally, what I would say is that the point of dark field is to increase contrast, and therefore it's the point that the colors are not real. You want to have a higher contrast than what you normally would be able to see. Right, so sometimes you deliberately do not want to have the real color simply in order to see it better. So that's a little bit a question. Yeah. Here's a fun fact: there exists something called the beard second. Yeah, I read about that, which is the distance the beard grows in a second. Yeah, that's and we determined now how many micrometers. It grows on a day. We can easily calculate how many in a second. Yeah. So the question is if the hair would look different in comparison. I would say yeah, everybody's hairs look different. There's a large variation, of course, and yeah, sure. I'm quite sure that there are certain differences. Just like yeah. Even there are differences in my own body. Different parts of my hair is from. But the question is always, and that's maybe the interesting one, is could it maybe be that there are more similarities than differences? This could also be a factor. Yeah? Could you identify somebody just by hair? I think I already mentioned that. Is what you always need is you need some kind of a reference, and it's a question of the sample size. I mean, if you've got, for example, I give you a simple example here. If you've got, I don't know. Blonde hair, very bright hair, and you know it must be from one of those 5 people. Maybe in a police investigation, a suspect, 5 suspects, and only one of them has actually a hair color like that. Then you, of course, can identify it. But I do not think that the variation is large enough to have a worldwide hair database and then simply compare and identify people by hair. I don't think so. That this is possible because also the hair is too different even in one person. Yeah? And what you probably would do is you do a DNA analysis. You would get some cells from the hair and do a DNA analysis and then compare that. Yeah. So this is basically, so the answer would be yes and no. Probably not by the hair itself, but probably by the DNA of the person that is on the hair. Yeah. Is it possible to take measurements using the cross table? I think this was the stage micrometer, right? Yes, it is possible, and I think I will take this as a, how do you say, as a hint or as a suggestion for a future video, okay, to show you how to use the micrometer of the stage to make measurements. Does it determine hair color? I also talked about this, at least in my white hair, gray hair that I had, the central part was still black. Okay, so to a certain extent, it does determine it, but we've also seen that the hair actually is pigmented throughout and not just in the center. Yeah. On the picture of the scale of the ruler, we can see an interesting regular pattern of the ink. Yeah, we can. I don't know if you just. Why not have a look again? Let's put it on here. Let me see. Let me see, where is the scope? Yeah. So that's you can actually see how the ruler, the ink was printed on the ruler, right? And that's actually also quite nice. Look, now we've got the hair and the ruler next to each other, right? And you cannot get both into focus, but you can all imagine it from over here to over here, in 1 millimeter. Maybe you're able to put 10 or 20 hair next to each other. Yeah. So this basically means that maybe about a tenth of a millimeter. That's actually something that's also coincidence here. You just basically put them together on a ruler, and then you can actually also make a comparison. Yeah? So let's go on. How thick is the medulla? Yeah, in humans, they say approximately a quarter of the total hair diameter. Yeah? How would you adjust parfocality? Basically, shims, these are basically little discs that you can put into the objectives. So, you know what, I'm just going to show this to you here as well. Parfocality means, just look at the one here at the top. Parfocality means it is when I switch from one magnification to the next one, that it's still in focus, right? And and if you use different objectives, oh my gosh, I found out. Look, now I know why it's not parfocal. Look, do you see this? It's not screwed in properly. Okay, I have to screw. Why does this become loose? I hope I'm turning this down to the correct direction. I don't know. I have to see. That's the reason. If it's not properly screwed in, if it's not properly screwed in, then of course it's too much down, and then you do not have. Then it's not parfocal. Yeah, so in other words, if you do not have objectives from the same series with a different focus, then you can adjust by inserting tiny discs where the objective, when you put in the objective. So if you put it all the way in, it cannot go all the way in, and this way you can adjust parfocality. Yeah, so yeah, this was actually was a little unexpected. Yeah, okay. Yeah. So now it's in here. Yeah, so I hope that this actually improves the situation. I don't know why it actually came loose. Yeah. So let's have a look here. What is the purpose of adding water? The purpose of adding water, for example, or mounting medium generally when I made those slides, is actually several. It's actually pretty important, the water. First of all, it prevents the specimen from becoming too thick, so the cover glass will actually press it flat. And second, it is important for the so-called refractive index. If you look at the specimen when it's surrounded by water, it will actually become more clear and more sharp and crisp. Yeah, and also it will appear brighter, and because if it's surrounded by air without water, then the air does not have the right refractive index, and then actually the objects will look too dark. Okay, and that's why water has multiple purposes. Yeah. Off topic, permanent slide. Is PVA glue a stable mounting medium, like is the one you can use for preserving specimens for 100 years? Honestly, PVA as a water-based mounting medium has been used for some time now. However, what I've been doing is I've been using just a PVA glue. I don't know how long stable it is. I know that those traditional microscope slides from the last century, they used a mounting medium very frequently called Canada balsam, and this has stood the test of time. It's over 100 years and it looks like new, right? So I do not know whether PVA glue also will last 100 years plus. Yeah, I would probably say, that would be my thing, is that this not homemade, I mean, this is just regular PVA glue which I bought from Amazon. It's not it's not official mounting medium, so to say. I would say that if you really want to make slides that last a very long time, you should then probably use laboratory grade PVA mounting medium or any laboratory grade mounting medium, because I don't know really what is, if they've added additives or I don't know. Yeah, so that's the thing. It's good for hobby use, good for hobby purposes. I've had good results with this for a couple of years. Whether it actually makes this slide stable for 100 years, I don't know. If you really want to make slides on a serious, how do you say, scale, then I probably would use mounting media that are established, if I may say it like that. Yeah. Can you explain why? So I think the question here with the bubbles is when you put hydrogen peroxide, H2O2, on a specimen, if you've seen my previous videos, like for example on pond water and so on, then there will be a lot of bubbles at the beginning. And the reason is because the hydrogen peroxide breaks down, forming oxygen. It reacts with the organic material, breaking it down, but it will also, because it's so unstable, the H2O2 will break down into oxygen and into water. The question is why does it do that so quickly? Because many cells will actually have an enzyme called catalase in it. I'll just write this down. Yeah? And this catalase enzyme is there to protect the cell from reactive oxygen species like H2O2. And basically the catalase will convert the H2O2, okay, to H2O and oxygen. Okay, you have to balance the equation. You probably want to do put a 2 over here, and you want to put a 2 over here. Okay, so 2 molecules of H2O2 are converted to 2 molecules of water and a molecule of oxygen. Yeah? And these are basically the bubbles that appear. Yeah? And the thing is that this is actually a sign that the H2O2 breaks down and that there is also H2O2 present. Yeah? So actually it's a good sign that something's going on. Yeah? And this basically means that very often there are bubbles. Yeah? And I have a question, is TPX mounting media, it contains xylene? Yes, I know. And I'm not sure if it would be dangerous to use because I'm not sure if xylene balsam is the same as xylene xylene. These xylenes are aromatic compounds, basically means ring-shaped compounds. And generally what I read is that if you can avoid it, you should avoid it. Okay, and I know, or I've heard that in hospitals, what they're using is when they have to make those slides, for example, of histological cross sections, they're using actually specifically solvents that do not contain xylene. So it's kind of a xylene free. Yeah, so they're kind of also advertising this because you kind of want to avoid it. Yeah, yeah. So that's the thing. I personally also prefer to avoid it because I'm doing this as a hobby and it's not necessary, and there are other safer alternatives. And yeah, for this reason, what I like to use is as a mounting medium, I like to use Uprol, kind of difficult to get. We've already talked about that. But you can simply then take the specimen from alcohol and put it into Uprol. And if you want to use, if you want to prepare specimens with xylene, like certain resin-based mounting media like DPX and even UKit, which I also have, contains solvents like xylene, then you have to make sure that the specimen is completely dehydrated and free of water, right? And that's already quite a lengthy process. So making those histological slides like the monkey tail that I have here, right? Yeah, like the monkey tail or the scalp, the human scalp, making these things is a lengthy process. Yeah, so I'll be honest with you, I've not been doing these things. Yeah, because simply it's yeah, you need a properly equipped lab. You have to dehydrate the whole thing. You need to do paraffin embedding. You need to, of course, microtoming is not so much the issue, but you want to stain it properly. You have to dehydrate it and so on. It takes time and the lab, and then at the end you have to put everything into xylene, and then yeah, and so that's why there are these more modern mounting media and solvents that do not contain that. Yeah? So what I'm going to do now is I'm going to quickly look at some of the, how do you say? Let's let's go back together in 21, 24 to examine those. Yeah. um, I just want to say the following. If you're interested I don't have the link ready here, but if you're interested in mounting media, I know that Microscopy UK, that's a pretty large website. They've got articles on mounting media, also recipes on how to make your own mounting media. I tried to make my own mounting media, but I have been not so successful. Because of the bubbles. You have to degas everything properly and so on. You need equipment. But essentially, polyvinyl alcohol and polyvinyl acetate, PVA, is a water-based mounting medium that has been used in the past quite a bit. Yeah. My skin looks just like that of onion under the microscope. Yeah, that's yeah. When you basically do the following, when you have a sunburn and you get the skin off, that's the top layer of the skin. Yeah. So, what's the purpose of adding water? I talked about this. Yeah. Do these onion cells have vacuoles? Yes, they do. Pretty large ones. And can they be made visible? You know what? Is the onion cell still in here? If you're asking already, then why not talk about those? Okay. Okay, this was again, I do not always want to redirect you to my other videos, but I did put onion cells under the microscope already before. And I'm just still redirecting you because actually those videos are better than what we see over here. And it's difficult to see because in the other videos I actually increased the contrast. But I'm just going to show you here. I'm just trying to find something a good one. It's so difficult to see, but you know what? I'm just going to use this onion cell over here. Yeah? What you see over here is that on the side, the question is, do they have a vacuole? What do you see over on the side here? It's a little bit, those dots on the side here, a little bit more. It seems to be the nucleus. This is where the cytoplasm is. Yeah. This dark line on the outside, that's the cell wall, and the whole thing on the inside here, that's the vacuole. Yeah. So it's huge. But if you really zoom in, then you're actually able to see that the vesicles, those little tiny dots, they're actually moving only on the outside here. So the cytoplasm of the cell is actually pressed really very far to the side of this towards the cell wall. Yeah. So now sometimes you're able to see. Look over here. You might see, but it cannot be because the nucleus over here is right in the middle. Right. It's not pressed to the side. Well, actually, it is pressed to the side, either on the top or on the bottom. Right, so it's still kind of more on the side, yeah. But we're not able to see it on the side because we're looking at it from the top. And if you look carefully right now, do you see all those little dots here? Yeah, that here these are all the vesicles, yeah. And then the vacuole is found beneath it, so to say, yeah. So it's because the onion cell is transparent. That's why it kind of everything looks like it's in one level. Yeah? So just for you a quick answer because of the question about the vacuole. Yeah, this was the one. I wasn't here from the beginning today. I still don't fully understand the difference between hair follicle and the hair root. Are the same? No, the hair follicle. Okay. Okay, good point. The hair root is actually the part of the hair which is inside the hair follicle. I don't know. Yeah. No, I don't have a hair root now. I'm just going to show you in a drawing. Okay, let's do it like this. If you've got, I don't know, yeah. If this is kind of the hair, then it's thicker than it's the hair root, but there are basically cells here around it, especially down here in the region down here, which are rapidly dividing. So there are even stem cells, adult stem cells in here. Right, which are making new cells. So that's basically on the outside. That is the follicle, right? And sometimes there are even and the actual hair is on the inside. Okay, and that we would say that this is kind of the hair root, and the follicle is basically on the outside. These are the cells that actually make the hair. Yeah? So that's very briefly. Yeah. So and yeah. I think that there are no more questions. Yeah, I think no matter how many... okay. No matter how many slices and names, the bigger, simpler picture is always lost on lab conditioned minds. Yes. Yeah. The monkey tail is like an all-in-one specimen, yes, because it contains all of the so many interesting things here. Yep, what am I gonna do now? It's almost 1.5 hours again. Okay. I purchased Canada balsam from Amazon. Really? They sell it on Amazon? Yeah. Oh, cool. Okay, interesting. Canada balsam, for those of you who don't know that, is a traditional mounting medium, which basically has been in the 19th century been used a lot, and is known for basically very long. If the specimen is properly dehydrated and so on, is known to be having very good long-term stability. Yeah. So I didn't know that. You can buy it on Amazon, because sometimes Amazon is a little bit restrictive in sending chemicals. But the specimen that you embed in Canada balsam has to be completely dry, also free of water, and that's usually always the case. Yeah. So I think I'm just going to, after almost 1.5 hours again, I'm going to almost call. Oh, look, the text is upside down. Yeah, call it quits again. What we do is the following. I'm just, for the fun of it, we're going back to the very beginning. For those of you who were missing out on the very beginning, I put my beard that I shaved under the microscope. And where is the scope? Here we go again. Okay. Let's go down a little bit. Look at the colors. Yeah. Okay. And what I just wanted to show you again, and this was in case you joined in late, this is how I started off. And by shaving yourself, you're getting a very good and unique view of the cross section of the hair, right? Yeah, like over here. And then you can see that the inside, that's the medulla, then you have the cortex, and on the outside here you have the cuticle. Yeah, it's an outside protective layer. Yeah? And that's actually one of the only ways that I know how to get without using some kind of embedding in the fancy microtomes, of actually getting a cross section here. Just wanted to share that with you. Okay. And I'm going to call it quits for today. Hope that you like it. I would like to invite you, please, still to subscribe to the channel. Maybe it would be kind of nice. It also kind of pushes the channel a little bit, makes it maybe a little bit more visible in YouTube. Right? If you have any suggestions, look, it's drying up already. If you have any suggestions on things that you want me to do, then please, mention them either by writing a comment. I've already promised you last week, and I'm working on it to make the pictures available of the sea urchin spines, the cross sections. Still working on it, and I'm also in the process of ordering glycerin gelatin, which is also a water-based mounting medium. And because this glycerin gelatin is pretty good for mounting pollen. I know it's now fall, autumn, so it's not pollen season. But if we figure out how this works, then when spring comes, then we're able to mount some pollen using glycerin gelatin according to Kisser. They're different recipes, and the one according to Kisser is the standard mounting medium used for pollen. So you can already see, for mounting different specimens, there are different mounting media that are recommended. Okay, enough for today. Okay, I wish you all the best. Happy microbe hunting as always. Watch the other videos in my channel as well. See you around next time. All the best, and bye-bye. Thank you that you've joined in again.